My name is Professor Dr. Chris Geddes. I'm a full professor, University of Maryland, UMBC campus. I presented and debuted the second generation LacriPen about 2 years ago at this great meeting. I'm here to introduce you our now third generation LacriPen. For those not familiar with the LacriPen is a portable handheld osmometer that can diagnose very rapidly within 1 second dry eye syndrome. It also has the ability to diagnose other different ocular disorders such as MMP9, herpes simplex, herpes zoster, methicillin-resistant Staph aureus, and a few others. We all know that osmolarity is really the single biophysical measurement that captures the balance of inputs and output flows within tear film dynamics, and hence it's really ideally suited, osmolarity, for the diagnosis of dry eye. As a community, we have known that osmolarity can diagnose dry eye for several decades. In fact, a lot of work by Farris, Tomlinson, and a few others has correlated the degree of tear osmolarity with the onset of dry eye syndrome. We all know that for a normal, healthy patient, the osmolarity of the eye is around 300 milliosmoles, and it can increase to as much as, say, 360 milliosmoles for very severe dry eye. What we perhaps don't all realize is that actually we need a device today that has a sensitivity of about 6 milliosmoles to accurately and reproducibly diagnose the different stages of dry eye, from normal to mildly to moderately and severely dry eye. Unfortunately, there is no device in the world today that's currently on the market that has a sensitivity of 6 milliosmoles or better to be able to do that. The LacriPen that I'm showing you can indeed do that. We have an analytical sensitivity of about 2 milliosmoles. So how does the LacriPen work? The LacriPen uses a technology called surface plasmon resonance. Light sources fall incident on the underside of a very thin gold film that actually touches the surface of the eye. It forms an image line on a sensor, a little black line, which we call the reflectivity minimum, and this little black line actually moves. And on the right-hand side, you can see a reflectivity minimum curve. That curve actually moves very minute amounts due to the tear osmolarity. And we've developed very complicated algorithms that can actually precisely determine where that reflectivity minimum is. Surface plasmon resonance has been around for about 30 years. In fact, I was recently in Singapore celebrating the 30-year anniversary of the discovery of SPR. And on the top left-hand corner, you'll see a typical commercial instrument. These things are about the size of washing machines and cost hundreds of thousands of dollars. LacriScience has spent many years on a marathon kind of development project, being able to condense this washing machine-sized technology down to a very smallable device that literally fits in the palm of your hand, and I'm holding one here. The key success to the LacriPen is really the sensor prism, which is at the tip of the pen, and this actually touches the eye and has that little gold filmed on. And that's where all the fancy optics are that allow us to do multi-angle, multi-wavelength, instantaneous diagnosis of dry eye syndrome. We have tested the LacriPen so far in salt solutions, in contrived artificial tears, as well as with a few clinical and veterinary patients. Here is a linear response to salt solutions. We can accurately accurately determine the level of salt in a salt solution in about 1 second with a sensitivity of about 2 milliosmoles. Here is some contrived tear data. We use 2 wavelengths in the LacriPen, an 855 and a 950 wavelengths. We use 2 wavelengths simultaneously so the pen will self-calibrate. We have linear responses in artificial tears in both channels. The measurements in tears are less than 1 second, so this is really an instantaneous measurement. You take the LacriPen, you just touch the eye, and you get an instantaneous readout of tear osmolarity. We have a current sensitivity of about 2 milliosmoles. Here is some patient data. You can see that the SPR minimum, that little black line, or the dip in the curve, it moves from a control waterline a few pixels for mildly dry eye, but then for more severe dry eye, you can see this move in the reflectivity minimum is more dramatic. Our algorithms that are located on board in the software within the LacriPen can actually determine the position of that and the exact degree of dry eye syndrome. In addition to dry eye, we've been able to develop sensors on these prisms for metalloprotease 9, MMP9. We can detect as low as 5 nanograms per milliliter of MMP9. We can do it instantaneously within a few seconds. That enables us to potentially monitor treatment downstream. There's no anesthesia needed and no scraping of the eyelid. It's important to know that there is a lateral flow assay on the market today that has a sensitivity about 40 nanograms per milliliter. It's important to note that the basal level in our eyes is 5 nanograms per milliliter. Clearly, the LacriPen is well positioned to address that. This is typically how we build up the biochemistry on our smart prisms. We have antibody-mediated detection of the metalloproteases by using dextran-coded slides. And because it's a platform technology, we can pretty much build any biochemistry for the detection of virtually any disorder in the eye. Of interest to us, of course, is MRSA, methicillin-resistant Staph aureus. We have immediate diagnosis. There's no waiting for cultures, no need for MRIs, no need for broad-spectrum antibiotics when using the LacriPen. You get instantaneous readout of what we have. Potentially huge savings for patient care as well. As I close down my talk, I'd like to stress really the LacriPen is a platform technology, not only an osmometer for determining dry eye syndrome, but we can detect adenovirus, no anesthesia needed, no scraping of the lid, especially for children where this is problematic. We're working on bacterial sensors of our little prisms so we can detect gram-positive and gram-negative bacteria more generally, instantaneously, as well as the LacriPen really has the potential to do a whole range of biomarkers, enzymes, and potentially other chemicals. I think the LacriPen is going to change the way we think and use ophthalmic devices. Thank you.